Mark, take it away.
Well, thank you, Godfrey. So I'm just going to share my screen, and hopefully this is going to work. So I hope you can all see that okay. Well, as Godfrey said, I've got quite a lot of interests, and I was very fortunate to have the paper selected by the editors of JMCC because they were perhaps struck by some of the new EM work that we had done. But in this seminar, I'd like to put that work in a slightly wider context. I'd like to tell you about the structures underlying cardiac EC coupling in a very brief way. I'm sure that's revision for most of you. I'm going to talk about some of the biophysical techniques we've used to try and analyze optically t-tubular structure and what we can deduce about how solutes might move inside t-tubules. And of course, because a major importance of understanding cardiac physiology is trying to understand the problem of heart failure, I'm going to try and put some of that into context as a basis to understand some of the changes that take place in heart failure. And then finally, I'm going to move into Dyad structure, the new Dyad structures we've seen using something called high pressure freezing, coupled with electron microscopic tomography. So that's sort of the outline of where we're going to go. Well, the structure, the microscopic structure or nanoscopic structure of cardiac muscle has been known for quite a while. The surface membrane, which carries the action potential, here colored in green, propagates in the working ventricular cell via things called t-tubules into the interior of the cell, where they cause calcium release from the sarcoplasmic reticulum. And although this picture is actually quite old, it's still actually perfectly accurate. This is a deduced structure by Fawcett and McNutt of cat myocardium. And if we look at thin sections of this structure in the cardiac muscle, which is unique to cardiac muscle, this is not what things look like in skeletal muscle. We see a dyadic junction, a junction between the sarcoplasmic reticulum here in blue and the surface membrane in green in the form here of a t-tubule. And this specialized structure, by analogy with skeletal muscle, was quickly recognized to be the source of cardiac excitation contraction coupling, because we knew that the sarcoplasmic reticulum released calcium, and we knew that the signal had to come from the action potential. So where the sarcoplasmic reticulum is in close contact with the t-tubule membrane is where signaling could take place. And so we can identify here junctions, the junctional sarcoplasmic reticulum, where it is closely opposed to the t-tubule membrane here in green. And between those two membrane structures, we have the so-called ryanodine receptors, which are extremely large proteins, which are in fact the calcium release channel of the sarcoplasmic reticulum and the source of calcium for the cell to contract. I'm sorry, I'm getting a bit of frog in my throat. So our view of EC coupling is this. It seems quite straightforward on the face of it. You have an action potential in the surface membrane of this cell. It propagates through the t-tubules, which are extensions of the surface membrane. And there it opens L-type calcium channels or dihydropyridine receptors. And this allows a little bit of calcium to come into the cell. And there a process called calcium-induced calcium release, as described by Alex Fabiato, takes place. And this causes these ryanodine receptor proteins to open, and they allow calcium to come out of the sarcoplasmic reticulum to raise calcium everywhere inside the cell and switch on the contractile machinery and cause the myocardial cell to contract. And we see that microscopic release in the form of these things that we call calcium sparks. So to understand how this machinery works and how it might go wrong in heart failure, we have to understand not just the biophysics and the molecular biology of the channels and proteins involved, but also understand the structure. And I like to use the analogy that, you know, proteins aren't alive, cells are alive. And the difference between the two is that it's the organization of the proteins that creates the living functional unit. Now, if we look at this junctional space where excitation contraction coupling takes place in more detail, perhaps here using electron tomography, so we can actually look carefully at the interrelationship between the proteins. A paper here in mouse by Hershey et al. The outline of the t-tubule membrane is in green, and the sarcoplasmic reticulum is in yellow. And you can see this very flattened pancake-type structure that in the mouse follows the t-tubules quite closely with extended axial junctions. This pancake structure wraps around a part of the t-tubule membrane, and in that space there are the ryanodine receptors and accessory proteins that are taking control of EC coupling. And we used, and Hershey et al. And other people use tomography because it allows you to see more carefully the interrelationship of the structures. And the most important point I'd like to drive home here is that the junctional sarcoplasmic reticulum, where it forms a junction with the t-tubule, appears like a rather flattened pancake structure of a size about comparable to this gap between the two membranes we call the dyadic gap. And it's there that the calcium comes across the t-tubule membrane and is released by the SR through ryanodine receptors. Now, if we look at human t-tubules by light microscopy, and this is a red-green anaglyph, so if you had some, it's rather old-fashioned technology, but if you had some RG glasses to put on, you could appreciate the structure in 3D. But you can see that in humans, the t-tubules are rather radial and spoke-like, rather unlike the rat and the mouse and other rodents. And you can see these spokes of t-tubules radiating into the interior of the cell, and in longitudinal selection, a lot of transverse tubules with far fewer axial connections than you see in rodents. So could we quantify anything more about how big these structures are, apart from using the very laborious method of electron microscopy that may introduce its own problems, as I'll describe in a minute? And we introduce a rather simple, quick method about which people would, maybe use. What you can do is you could stain the surface membrane of the cell with a suitable membrane marker, here FM464, and you can also fill the interior of the tubules with a marker, here calcine. So what you can see there is a measure of the surface membrane of the cell and the interior volume of the cell. And so the ratio of volume to surface area must be related to the size of the underlying structure. So we calibrate this volume to surface area ratio, and that's seen here in the mouse. And you can see that in various regions of this T-tubular structure, the volume to surface area varies, and we can quantify that in cells in this histogram. When we look at the rabbit, which looks more like human with more radial-like spokes, we can see a rather different volume to surface area ratio through the cell. In fact, generally speaking, the rabbits have more volume per unit surface area. Well, if we make some assumptions, such as that the shape of the underlying structure is constant, here we assume it's roughly cylindrical or close to being a cylinder, then in fact we can calibrate those signals based upon the labeling at the surface and in the bulk solution. And we end up with a rather simple formula that relates the diameter of the T-tubule to this volume to surface area ratio. And this paper is in a journal Molecular and Cellular Cardiology if you're interested in looking into the methods a bit more. And interestingly, this Vita-SA method, which gives a very quick way of surveying a very large amount of data inside multiple cells as compared to electron microscopy, agreed rather well with our electron microscopic measurements of T-tubule width. And generally, the rabbit T-tubules, on average, are about twice as wide as mouse tubules. And that has important implications because from this, we can calculate the so-called electrical space constant of the T-tubule and think about how well coupled it actually is. And it turns out the electrical space constant is much bigger than the radius or length of the cell. So T-tubules should be well quantified. But what about entry of solutes? Well, we can use the same trick. We can put the cell in different molecular weight solutes. We can calibrate the size of those solutes using fluorescence correlation spectroscopy, and that's shown in this lower graph. And we can look at how well they go in. And the surprising thing was that low molecular weight solutes go in quite well, but we appeared to get a cutoff at higher molecular weights around the size of proteins. So proteins would tend to be apparently excluded from the t-tubule lemma, which is actually a little bit of a surprise because the radius here is less than 10 nanometers, but the t-tubule radius is about, or width is about 300 nanometers. So something is restricting the entry of proteins perhaps into the t-tubule lumen, and we don't know quite why that is. Now, early electrical measurements have suggested that indeed even potassium was restricted in its diffusion into the T system, and some later experiments using an optical technique called FRAP supported that idea, and in fact suggested it was even more restricted. I just want to touch on what FRAP is for a moment. The idea is that you bathe the cell in a fluorescent medium, and then you bleach the fluorescence inside the cell using a confocal microscope or a bright light source. And then having bleached the fluorescence inside the t-tubules, you watch it recover. And that recovery must reflect the diffusion of fresh dye into t-tubules. What's interesting about these experiments by Scardigli and Sacconi's group is that they saw that in heart failure, there was more restricted diffusion in the t-tubules. Well, clearly, this has potential implications for solute exchange, and also because the electrical signals are related to the free movement of solutes, such as potassium in the T system. That could have implications. And one of the interesting things that Sacconi's group showed was that in heart failure preparation, the action potential appeared to fail in some parts of the T-tubule. And obviously this has big knock-on effects for excitation contraction coupling if you have an AP failure inside the T-tubule membrane. At the same time that Sacconi's work was being done, we were actually using the same techniques, and we're trying to struggle with the idea of FRAP. And the problem for my group thinking about it was that it's actually rather more complicated than it seems. It isn't just a question of bleaching and looking something coming in. We have a rather complicated situation where we have flow with restricted flow near surfaces that alter the pattern at which the dye can be replaced. And you can see here there's a cell, a typical cell above a cover slip, and this shows the velocity of the flow around that cell. It's done with solving the Navier-Stokes equations. So when we couple that with fluorescence recovery after photobleaching, which we're showing here simulated at the bottom left, we can photolyze the dye and see the dye concentration fall, but it doesn't fall uniformly around the cell. And this, we believe, will affect the signals that we see. So we may not be able to measure so easily exactly how solutes are moving with this type of experiment. And we can sort of summarize that even with the highest flow rates that we could achieve without actually washing cells away, which is about 2 millimeters a second across the surface, we still were in a situation where there was surface effects limiting solution exchange around the cell. An alternative is to have no flow, and in that case, the whole problem actually becomes simpler. We don't have to use such complicated fluid dynamics to actually try to analyze what might be going on, and which would be different for every single experiment, because the flow pattern would be different, the cell would be different, etc. We could just simply immerse cells in bath and allow diffusion everywhere to take place. And when we did that, we could see this sort of pseudo-exponential recovery of fluorescence inside the cell. And we could computer fit that to ask what the diffusion coefficient is inside the T-tubular network that's present inside the cell. And the computer could fit this extremely well for such networks inside rabbit and mouse, at least in general. However, something strange appeared when we looked at the transverse gradients, that's the gradients across the width of the cell, that in fact there was a systematic error, seen here as the difference in red and the gray trace. There was a systematic error in the solution that could be removed by supposing that there was a barrier at the surface of the cell selectively. And that barrier within the first micron or so of the cell enabled then the computer to fit our FRAP data much more accurately, and the residuals are shown in the lower part. And when we looked with electron microscopy at the surface of the cell, looking at the t-tubule mouths, we could see that the rabbit, where we didn't need a barrier, looked like it was sort of continuous with the extracellular space. There's quite a neat mouth to it, highlighted here. And when we look in a 3D rendering of a tomogram, we can see it looks like a tubular entry into the interior of the cell. When we looked at mice, the entrance was much harder to distinguish. There was all sorts of electron densities in the mouths, and it's very difficult to see this very complicated t-tubule entry into the interior of the cell. And here it is rendered, and I think you can appreciate that this structure could easily be the explanation for why we appeared to need restricted entry into the interior of the cell. So there are all sorts of structural effects taking place, and there is the effect of ellipticity, which we can measure inside the cell. But by using the images of the T system, coupled with computer modeling, we could assign the relative contributions of all the morphometric effects that we're interested in to the effects on solute transport. So we could look at the tortuosity, which is how non-linear the tubules are. We could look at the effect of longitudinal tubules, which are relatively less common in human and rabbit and more common in mouse and rat. Varicosities, which are local variations in the size of the diameter of the tubule, and also the effect of this surface membrane barrier. And when we multiply all these effects together, we predict by how much the solute transfer might be reduced. And you can see that these factors, when multiplied up, appear sufficient to more or less explain the bulk FRAP data which we had obtained in these experiments. What about heart failure? Well, heart failure is a massive problem, as you can see here, affecting a very large number of people, particularly as you age. And in heart failure, we know there are lots and lots of things going on. And we're particularly interested in the tissue, cellular, and subcellular remodeling, as well as some electrical and gene expression changes that take place in the cell. And you'd have to have a whole seminar series to even touch on some of the things that change in heart failure. But I just want to talk about subcellular remodeling at the moment and just introduce you to that. So these are tagged cine MRI images from which we can deduce local contraction inside heart failure patients. And we actually were able to get these hearts at explant. For sampling, we developed techniques that would enable us to sample them. And when we look at two t-tubules of heart failure, we see that in the normal cell, it's reasonably linear, while in the heart failure cell with strong contraction, which those are regions of the heart that are contracting well as measured by tagged cine MRI, the structure is more or less uniform, but starting to show some changes. When we look at regions that don't contract, the t-tubules are heavily damaged, mostly missing. And in fact, this isn't relating to underlying sarcomere structure. The sarcomeres are labeled here in red. So t-tubule membranes are changed in heart failure. And even within one heart, we can see regional variations in contraction strength. And using this tagging method, coupled with a computer-aided analysis, we can identify different regions which contract strongly or weakly. On average, of course, the failing heart contracts quite weakly. And we quantify this as the circumferential shortening. So in this failing heart here, the green trace, in the left ventricle, it's contracting fairly well. The septum is some regions, the left ventricle, other regions are not contracting well. And here the septum isn't contracting at all. In fact, it's being stretched during systole. And that can be compared to normal. So if we look at analysis now of how well the cell contracts compared to the organization of t-tubules, we see a correlation. We used all sorts of measures for t-tubule organization, such as the power of the Fourier transform of the image, et cetera, But the strongest correlation was seen when we considered the angle of the t-tubules to cell axis. When the transverse tubules were not transverse so much anymore, then we see the greatest diminution of contractile performance. And here in this slide, normal hearts are in black, and we assume they have 20% circumferential contraction. We couldn't actually, of course, MRI these normal hearts because these were road traffic accidents and the hearts that came in. So there was no chance to MRI them before the hearts were made available for analysis. And each of these dots is an individual patient. So you can see that even within one heart, we have a correlation with the organization of the T system. Now, this disorganization of the T system isn't completely new. It had been shown in a rat model by Pies-Cheng's group that in a heart failure model, there was a loss of transversality of the T system to become more longitudinal. And there was also a reduction in co-localization between the dihydropyridine receptors, the L-type calcium channels in the surface membrane, and the SR ryanodine receptors. And interestingly, in our human samples, we saw something extremely similar, a loss of co-localization. So it isn't that you've lost your calcium channels that trigger calcium release from the sarcoplasmic reticulum. In fact, just a large number of them become misplaced and not quite in the right place anymore. So something has happened for trafficking that we need to understand. Which brings me now to the subject matter that JMCC thought was interesting enough to invite me to give you this seminar. And it's about high pressure freezing, a way of doing electron microscopy in a slightly different way. The idea is to use fixation of preparation without dehydration and strong chemical fixatives such as glutaraldehyde that cross-link proteins. And we do that by trapping the tissue in amorphous ice at high pressure very, very quickly. And you can see the structures of water here on the right-hand side. You have stable water, and as you cool it rapidly, you go through supercooled water into a region where you can have homogeneous nucleation. That means that ice crystals are forming everywhere inside the preparation. But at high pressures, you get amorphous ice, ice with no structure. And that's because the ice is not allowed to expand due to the high pressure. And so we move into glassy ice forms down here. A record from the machine here on the left that does it, a Leica EM-PAC2 machine, is shown here. And then you can see the pressure rising extremely rapidly on the specimen, up to over 2,000 bars, which is the equivalent to 0.2 gigapascals. And at the same time, just after the pressure has nearly reached 2,000 bar, it's rapidly cooled by liquid nitrogen down to minus 195 at a rate of 24,000 degrees per second. So this machine essentially traps the structures in its carriers in amorphous ice in a matter of a millisecond or so. So there's no possibility of any ice formation, no disruption of structure. We've trapped all the structures in amorphous ice, and then we process it to look at it in the EM. And I don't want to go into all the details, but the key points here is that the carriers that enable us to do this are very, very small. They're 1.5 millimeters in diameter by 0.1 millimeter deep. And once we've frozen it with liquid nitrogen, we do something called freeze substitution with osmium tetroxide and uranyl acetate in acetone at minus 90. The amorphous ice doesn't melt, it's still amorphous. And that then fixes the membranes where they are. And then we use EPON processing to look at the specimens. So this is the result. You can see some structures inside here. On the right, the tomogram's just running through a section of the specimen. Of course, the mitochondria with their cristae. You can see transverse tubules, t-tubules. This is a rabbit. And we can also see the sarcoplasmic reticulum. And the surprising thing about this data, and which was a little bit of a shock to us, was that the structure looked really quite different. Instead of these very thin pancakes, whose thickness is comparable to that of the dyadic space, they appeared more inflated and more pillow-like, much more rounded membranes around the T system. And you can see that that isn't just because the plane and section. Everywhere you look in the section, you see similar things going on. So if we look in a bit more detail inside there, so by conventional glutaraldehyde fixation, we too see collapsed junctional membranes, junctional sarcoplasmic reticulum membranes in the dyad structure. But with high pressure freezing, these structures are much more inflated and pillow-like with smoother membranes. And you can also see the gap between the T-tubule membrane and the SR membrane isn't constant. It varies from spot to spot. The data is summarized here if we measure with a tomogram, from the tomogram, so we can measure orthogonally from the surface. We can actually measure the exact height, the junction. We can see here that the junctional width is seen with high pressure freezing is much larger than for glutaraldehyde fixation. And we see essentially the same thing in mouse. And there's some renderings here of the membranes in blue. This is the junctional sarcoplasmic reticulum in blue by high pressure freezing. And it looks quite different from the chemical fixation method. Now, why we should be interested in that junctional space is because it's the key to how EC coupling works. Inside that junctional space, the calcium that comes in through the dihydropyridine receptors here in black causes a large increase in local calcium. It raises local calcium by a factor of more than 100. And that enables relatively insensitive ryanodine receptors to open and release even more calcium in this calcium-induced calcium release process. So the geometry here is what enables the system to have very high fidelity in response to an action potential and still at the same time be quite stable at rest and not cause abnormal calcium release. And similarly, the termination of calcium-induced calcium release, we believe is mostly due to something that we've called induction decay, and it's due to depletion of calcium inside this junction, causing the flux through ryanodine receptors to decrease. So the geometry of this structure feeds into the efficiency of EC coupling as well as its termination. And when we look at the width of the dyadic gap inside our high pressure frozen samples, here in blue, we see that it looks rather different from the tissue or cell structures. We see a wider range of distances inside the dyad. It's not quite as uniform as we thought it was originally. And in some places, it can oppose much more closely. So chemical fixation appears to make the dyad gap a little bit wider and perhaps more uniform than what we see with high pressure freezing. And even more, inside the sarcoplasmic reticulum, now it's not a collapsed structure, we can even see electron densities that may be proteins. And we think this could be perhaps polymerized calsequestrin. Now, while calsequestrin isn't thought to be changed very much in heart failure, there is some evidence here cited by Karash et al. A couple of years ago, that there may be changes in glycosylation and phosphorylation calsequestrin that may affect the trafficking of calsequestrin and may affect the formation of these dyad structures. So they invoke the phosphorylation of calsequestrin in trafficking and formation of the dyads. And we know that in heart failure, we appear to be seeing dyads being lost, and they may also break up, as some evidence from Bill Lauch's group that point in that general direction. And finally, the network SR also looks different. Here on the right, by high pressure freezing, network SR membranes look beautifully smooth and rounded, and slightly wider diameter than we had thought. And of course, it's the network SR that takes up calcium inside the cell. So to summarize, I'd like you to think that we can get more than just pretty pictures by using confocal microscopes. We can actually get real quantitative data with a bit of creative image processing, and that there are marked differences that we can see in the T-tubular structure between rodents and humans and rats. Rodents, humans, and rabbits. Rabbit is much more like human in our hands. And in human heart failure, we lose T-tubules, and that's strongly correlated with the defects in EC coupling. And some of my other work, which I'm not talking about here, relates some of the effects of this on something we call late calcium spark production, which we believe is arrhythmogenic. And finally, the high pressure freezing method that we've applied for the first time to cardiac muscle suggests that the junctional SR and the network SR are not quite as we thought they were. And we obviously clearly need to do more work to make sense of this. And I'd like to thank everyone who's contributed to this, particularly the people who've donated money and facilities for us to use. particularly at Bristol, Cherry Kong, who's heavily been involved in the optical work, and Eva Rodzelenka, who came to Bristol to work with us on some high pressure freezing, and particularly Paul Vacade, Judith Mantell, and Ginny Tilley, who took the time to teach me how to use an electron microscope and try and do this sort of stuff. And Clive Orchard, who was supporting the group through its looking at FRAP and calcium movements. And in Freiburg, Peter Cole's group, where Ava now is, were instrumental at supplying a lot of the data analysis about t-tubule structures that we've seen. And finally, going back to the very early work on human t-tubules by light microscopy, David Crossman, Christian Soller, and Alistair Young, whose paper you might like to look at how we developed a registration system to enable us to sample explanted human hearts for regional differences in protein expression, as well as actually t-tubule structure. And with that, I'd like to say thank you very much for everyone listening.
Good. Thank you very much, Mark. It's a shame we can't have an applause button that we can press, but that was just to reflect how excellent that talk was. So we've got questions. They actually are, I see a couple in the Q&A, but there's actually the majority of them come through the chat format. So let me try and represent these. So let me try and have a go at this. Yep, so it's from Emma Robbins. She says, I'm an epigeneticist, so trying to keep up with EFIS, and asks, do you see any differences in t-tubule structure in heart failure with reduced ejection fraction versus heart failure with preserved ejection fraction in human hearts relative to normal hearts?
We haven't had a large number of different types of heart failure, and all that we know at this point, and all that I can tell you because the limited number of samples that we've had, is that in all cases we see disruptions in t-tubule structure. Now, whether there are subtle differences with regard to, for example, whether or not the junctions break up in the same way, or the t-tubules are as angled, I can't say, because as you can probably imagine, it's quite, well, when we were doing this study, the number of patients that we could get them fully MRI'd was actually quite low. And then to be able to guarantee that we could get the hearts in a timely way at the time of transplantation, the explanted heart, was problematic. But I think this technique could perhaps answer that type of question if you can combine the MRI tagging with your then subsequent sampling. These are questions I think you could really address quite well. So in answer, no, but I'm pretty sure you'll find the t-tubules are disrupted.
Thanks, Mark. Now McQuaid has answered Emma and directed it towards a Frisk et al. Paper in JMCC at 2021. So that's thanks now for doing that. Daniel Johnson asks, beautiful presentation and very interesting technique. Do you think that up until now we've therefore been under or overestimating the number of non-coupled RYRs given? As using this new technique, it also appears that there may be more coupled RYRs. Have you done any staining using this technique for other SR proteins, such as, what's that, HRC? Yes. Yep.
I'd have to say that this work is very much at its very early stages. Although we started doing high pressure freezing quite a few years ago, we were actually quite worried about what we were seeing here and whether or not this was really reflecting the true structure. We tried doing various control experiments to address that question. For example, why we tried doing some tissue, and it proved extremely difficult to get the microscopic biopsies that you would need to actually go in and do that. It's a bit of an upset for the electron microscopy field to think that the severe shrinkage that you see in glutaraldehyde might not be spatially uniform across the cell, and in fact could change the general appearance of the structures. I mean, I think the reason why we've seen the t-tubule junctional membrane inflated is because the calsequestrin and the other proteins in the JSR are not shrinking together. And that doesn't just make the thing a pancake, but it also pulls the t-tubule membrane away from the the t-tubule membrane away from the junctional SR membrane, except where there is also a lot of protein. So you're getting disproportionate forces inside the junctional SR. And so we're seeing, these changes in distance. And the biggest problem that we've got is that because we're using just osmium and uranyl acetate, we don't have huge contrast to be able to see the ranitidine receptors. That doesn't mean to say it can't be done. It's just that they're harder to spot in there than perhaps is the case for glutaraldehyde fixation. I'll be perfectly honest with you. I mean, I'm a complete beginner when it comes to electron microscopy, and I'm sure that there are people who are much more skilled than myself and my group looking at this. And we can only hope that we can move this forward with some funding to try and look at these sort of questions and actually try to quantify the ranitidine receptors that right now haven't jumped off the page at me when I've been looking at these tomograms. So do I think we're going to see big changes? I think what we would like to be able to do is to look at failing samples, failing cells. We've actually frozen some, but we haven't had the money yet to put them into the electron microscope. And actually look if we can actually ask questions about this question about, do the ranitidine receptor subclusters break up? The idea is that ranitidine receptors, we don't think they're in plaques anymore with polycrystalline type structure. We think there are small groups making up what we call a supercluster, that is the calcium release unit. And what I'm particularly interested in is, are those subclusters all connected through one bit of pillow-like junctional sarcoplasmic reticulum, or is it several bits being connected through the network? And I think that is quite a key question. So we have the samples, we don't have the funding, and we've been locked down for the past more than 12 months because of this COVID thing. But maybe in a year or so, we might have more insight into what goes on in heart failure, at least in heart failure rabbits. So that's all I can say at this point.
Good, thanks, Mark. Llewellyn Roderick, hi Llewellyn. He asks, I assume that most of the data is generated in quiescent relaxed cardiac myocytes. When contracting, do you see facilitation of solute movement also is contracted? In contracted myocytes, does the shape of the dyad change? Which is the point.
Oh, that's a really good question and a really hard experiment to do. Although the high pressure freezing is incredibly quick, the problem is that it takes place in a microcarrier that's really, really small. And we can put the cells in it, quiescent cells. I wouldn't want to put the cells in completely depolarized because that's kind of abnormal. The cells would be in a complete contracture. So I don't think we'd want to do that. I mean, it would be great because it's so fast that in principle, you could trap a cell in that state, in a contracting state. In principle, it's just I don't know how to do it. I wouldn't know how to electrically stimulate the cell just before the carrier flies into the freezing chamber to be pressurized. But you could certainly imagine that might be possible because the machine does it in about 10 milliseconds. So, you could certainly do it during contraction cycle. If you just work out how to electrically stimulate the cell inside that carrier, that'd be a pretty amazing thing to do.
So can I just jump in here? I know that Mike Shattuck is on the line, and I know he was doing similar things with freezing at a contraction point of a working Langendorf perfused heart. So maybe Mike can give his opinions on this.
Yeah, I mean, yes, that's absolutely right. I mean, you could smash the tissue flat with, aluminium blocks. I mean, that was first done in skeletal muscle many, many years ago. And yeah, you could definitely do that. The problem you've got is you haven't gone to amorphous ice. And that's really what you want.
Yep. Okay, we've got tons of questions, so let's get through them. So from John McCrill, I think, really interesting ideas. Is there any evidence on the molecular nature of the t-tubule barrier? The one you mentioned in mice?
No, I mean, I think it's part of the basement membrane, but in the mouse just is just, well, I think, I mean, in the EM, it looks like, basic membrane, like so proteoglycan type material, on the surface of the cell. What makes the t-tubule so invaginated, though, is more of an unknown. I don't know why that is. Because you can also see protoglycans in the rabbit as well. It's just not as obstructive because the basic t-tubule is much wider. So the answer is no, I don't know what the protein is, but with these techniques, of course, you can do correlative microscopy. And I mean, we haven't even started to do that sort of thing yet. Once these things are frozen, in principle, you can start to look at protein labeling and other things like that to, try and ask that sort of question. We just haven't got there.
Good. I'm going to switch quickly over, if you don't mind, to some of the Q&As because I realize that they've been piling up as well. And Glenn Tibbetts, great talk. The dyadic structure is interesting. Does HPF freezing give you a detailed structure of the sarcomeres as well? Looking at images, there appear to be differences in line filaments. Did you notice anything?
No, the basic structure looks pretty similar. It's just that the overall level of protein staining is lower in the experiments we've done because it's just how it turns out with these thick sections for tomography and moderately low levels of urinal acetate with osmium. You can't see such filament, but where we can see the filaments, the A bands and the I bands are as you'd expect lengthwise from the physiology. Sarcomere spacing looks about right. It's just you can't see, the filaments are just not so darkly stained.
Okay, next one, Josh Goldhaber. Hi, Josh. Mark says, Mark, hello, this is beautiful work. Can you comment on what Fig 3 of your paper is, the colorized blue and green membrane of the junctional SR and T-tubules? What are the folds? He's asking what they are exactly.
Yeah, well, this pillow isn't just an inflated balloon. It does also have, some soft, soft pillow-like structure in it. I mean, there are things, that are pushing probably on that balloon, if I could call it, or that pillow. So yes, they're not so much folds, but extensions of the network SR and also just variations in the general shape of the pillow. Mm-hmm. So I mean, I'm aware of the fact that maybe the way that that's presented in the paper, it's a bit hard because it's just, there are just lines traced from individual sections that are being calculated in the tomogram. And so it produces, rather than a 3D structure, a stack of lines that might be a bit difficult to interpret in places. But no, it's not internally folded as far as I can remember.
Great, just switching over to chat quickly. Mishra Abhishek, I think, asks whether you've done any of this work in Purkinje fibers and can see any difference to working heart cells.
Unfortunately, no, we haven't done it in Purkinje fibers.
No. And do you think that junctophyllin, I think this, or GPH2 can still bridge the junction in your revised structure?
Yes, I think it does. I mean, in fact, it's easier in some places because we're seeing a narrower gap. It's even easier for junctophyllin. I mean, it's perhaps attractive to think that, the giant ryanodine receptor structure will tend to keep those membranes apart by the size of the ryanodine receptor head, which I think is about 12 or 13 nanometers high. And the junctophyllin is trying to pull the membranes together. Well, that's what we think the structure, I mean, from the knockout experiment showing that the Dyad gap opens up if you get decreased expression of junctophyllin. I mean, you could imagine that regions where you don't have the ryanodine receptors pushing it apart, the membranes might naturally tend to spring a little bit closer together. So if anything, the fact that we see closer distances makes it easier for junctophyllin to weld the two membranes opposite each other. Right,
good. Mike Shattuck has a technical question about the FRAP experiments. Did you allow for bleaching of the fluorophore in the solution above the T-tubule? This is,
yeah, absolutely, yeah. If you look at the whole, I mean, I could go back to the image, but yes, the whole hourglass-shaped bleaching structure that is produced by the microscope objective is included in the calculation. So yes, we include it above and below.
Okay, another question from Mike Kaplikov. Beautiful talk. Given the differences in EM images provided by the different preparation techniques and the difficulty in preparing primary myocyte samples, such as that preparations might be of different quality between species, how do you evaluate which of the results are physiologically relevant? Oh.
Okay, so the first thing is that we fixed cells both chemically and by high pressure freezing from the same batch. And we only look at cells that appear to have normal resting sarcomere spacings. There's no evidence of any hypercontracture. There are no I mean, when you look at a living cell on the microscope, you see blebs and things appearing, vacuoles appearing if there's damage to the cell. So any cell that looked anything like that was just automatically rejected from the analysis. Now, does that mean that the high pressure frozen cells are worse than chemically fixed? Well, I don't think so, because the very nature of trapping the tissue in amorphous ice in a matter of a few milliseconds surely means that the membranes should be trapped in a closer to physiological state than if we wait for a strong chemical fixative to diffuse through the cell and fix structures as it diffuses across the membrane. So the freezing wave travels much, much faster. In fact, it should travel at the speed of sound compared to the diffusion wave and the diffusion and chemical wave associated with glutaraldehyde. So I think everything argues that high pressure freezing should be associated with fewer artifacts. But of course, we have no living gold standard that we could actually say, this is what the cell actually looks like. All that we know is that the t-tubules that we measure agree very well with our optical measures inside living cells. I mean, we see the correct diameters, as you saw in that histogram. The agreement was incredibly tight. So the high pressure freezing by itself doesn't seem to upset the t-tubules. But of course, I can't be sure that the SR hasn't, developed some strange properties.
Great. From Gulam Gilbert, thanks for the nice talk. Does the change in t-tubule orientation in heart failure, is it also linked to the change in SR junction with of the SR? It's a pancake shape. How does the new t-tubule interact with the SR?
We don't know. And that's what I say. We have some samples frozen. We haven't processed them and analyzed them yet. I'm sorry, I can't answer your question right now. Maybe we'll know in the not too distant future.
Okay, I've got a question. I'm going to try and squeeze in, Mark. Calsequestrin, you referred to it in the terminal cisternae as being polymerized. Yep. Mm-hmm. So what I'm concerned about is what do you think that polymerization reaction means for buffering in that section of the terminal cisternae? Because obviously it's where do you think it starts? At the it's multiply polymerized in diastole, I guess, to store. But during systole, you imagine the polymerization falls away. The change in buffering that incurs, does that shape in polymerization change the SR volume potentially? The lack of polymerization and the buffering changes that occur, how does that affect release through the randagene receptor? Do you know? Well,
I mean, I think you're going back to an interesting idea that was proposed by Eduardo Rios and his group a little while ago, where they thought that the polymerization, depolymerization of calsequestrin could be really important in determining free calcium levels. And it's the free calcium level, not the bound calcium level, that determines the flux through a ranitidine receptor. So yeah, I mean, it is possible that there could be dynamic polymerization, depolymerization inside that junction. And I suppose it might be fanciful to think, well, crikey, if those threads of calsequestrin that in some cases seem to go from one side of the pillow to the other, if they break up, maybe the pillow could change structure. And I mean, I wouldn't go so far to say as it would create a pump for calcium. I think that might be a bit far-fetched, because the bulk motion of fluid would be highly problematic inside that space. It's certainly interesting to think about, well, if the affinities of the binding proteins start to change, this might have knock-on effects, both for the repriming of calcium release inside the junction, as well as possibly shaping the terminal phases of the release process. So all I can say is they are all sorts of intriguing conjectures that one could make, Godfrey, about, you know, whether or not this might have severe functional knock-on effects. But yeah, anything is possible, I guess.
Sure. Thanks, Mark. The from Parisa Ashgrari, great talk here. Have you seen changes in cardiac sarcomere length in the samples compared to in the high pressure versus the normal fixation?
Yeah, the sarcomere length in the high pressure frozen is longer. We typically see about 1.7 to 1.8 micron sarcomere length. There is, in fact, the resting sarcomere length that we see in unfixed living cells, whereas in glutaraldehyde fixation, the sarcomere length is shorter, about 20% or so.
Okay, thank you very much. I mean, it's been a fantastic talk and a great session of questions. I really just have to say thank you again, Mark, for a really lovely talk featuring a nice paper, but also this additional work you've shown us has really intrigued us. And as always, the interesting stuff about your work is it brings up just as many questions as it solves, and spurs us on to do more work. Thank you very much, Mark.
Oh, thank you, Godfrey. I wish I could answer more questions.
May I just thank both of you, Mark, for a really wonderful talk, but also for getting through all these questions. There were quite a few, and all within an hour as well. So I think you have impeccable timing. Actually, in fact, the best timing I've seen throughout the webinar series. So well done.
Well, thank you. You're too generous, Davor. Thank you. It's been
thank you.
Fun.
See you for the next webinar, everyone.
Okay.
Cheers.
Thank you, everybody.
Thank you.
Bye-bye.